In this video, we're going to talk about the mechanism of smooth muscle contraction and relaxation. So I'm just going to draw sort of a generic cell membrane here for smooth muscle. And inside of here, we are going to put a sarcoplasmic reticulum. I'm just going to label that SR. And we know that our sarcoplasmic reticulum is responsible for storing calcium. Okay. And then on the cell membrane, I am going to put a calcium channel. Okay, and then I'm going to put a calcium channel on the sarcoplasmic reticulum. Okay, so it kind of looks similar to skeletal muscle, but notice that up here I've got a calcium channel and I've got a calcium channel here. So it's starting to see a little bit of difference. So in smooth muscle, the first thing that's going to happen is that this calcium channel is going to open up and calcium is going to flow into the cell. Now, what causes this calcium channel to open up is a variance on, could be a ligand from a hormone, it could be a G protein coupled receptor, it could be an action potential, right? But something's going to open up this calcium channel. Calcium is going to come down and it's going to bind to this channel here, causing it to open up and calcium to flow from the sarcoplasmic reticulum into the sarcoplasm. Okay, so we get two sources of calcium in the smooth muscle. So it's a little bit different than what we see in skeletal muscle. Now this calcium is going to come over and it's going to bind to a molecule called calmodulin. And calmodulin is just a generic class of calcium regulated modulators, right? So calcium will come over and it's going to bind to that calmodulin. Now, calmodulin inside of smooth muscle can do a variety of different things, but one of the major things that it's going to do is it's going to take an inactive molecule of myosin light chain kinase. I'm going to abbreviate MLCK, myosin light chain kinase. Right? And it's going to convert it over to an active form. So we'll call this AMLCK. Right? And I'll just write up here that this is myosin light chain kinase. Okay, so that's our myosin light chain kinase. Now, this myosin light chain kinase, right, is a kinase. So what does it do? It phosphorylates something. So what is it going to phosphorylate? It's going to phosphorylate myosin. So I'm going to draw sort of our myosin head down here like so. Now remember, myosin is going to go up, and myosin will bind to an actin filament. And this is where things get a little bit tricky with smooth muscle. Okay, now smooth muscle does have a molecule of troponin that surrounds it. So we'll put this in, let's do gray here. Right, so it does have tropomyosin that sort of wraps around it, just like it does in the skeletal muscle. And attached to it are two molecules. One is calponin, and the other is something known as caldesmin, right? And so there is some evidence that suggests this myosin light chain kinase will come down and it sort of binds to this, right, which causes a conformational change. And there's some evidence that says this calmodulin also comes down and it binds to that caldesmin and causes a conformational change, right? And there's a little bit of evidence that suggests that this tropomyosin moves. However, this tropomyosin does not, right, this tropomyosin here does not block the myosin binding sites in the same way that it does in skeletal muscle. So what really happens is this calmodulin comes down and it phosphorylates that myosin head, right? It adds a molecule of ATP to it. So myosin cannot bind to the actin filament until it becomes phosphorylated. So once we add that molecule of ATP, that myosin head will come up and it will bind to that actin filament. And it's the activation of these two molecules here, caldesmin and calponin, that allow that ATP to be hydrolyzed into ADP and inorganic phosphate, which favors that sort of binding. Okay, so it will get hydrolyzed into ADP an inorganic phosphate. So I'm just sort of draw this over here, what that would look like, right? Just so we can see it, is that we'll have ADP plus our inorganic phosphate when it's actually bound here. All right, so little bit different on sort of what happens in order to get this myosin head to bind to the actin filament. All right, so now let's just go ahead and let's write out some steps for this. Okay, so step one, right, is that there is a signal that causes calcium influx from the extracellular environment. So we'll put ECF here. Okay, and I just put signal because we don't really know what its signal is. It can be different in different scenarios. Okay, right, so the calcium, right, is going to cause or causes more calcium release from the sarcoplasmic reticulum. Right? Then we have our calcium activates calmodulin. Right? And then once calmodulin is activated, right, calmodulin activates, so calmodulin, we'll abbreviate that CAM, activates that myosin light chain kinase, so MLCK. Okay, so the MLCK, myosin light chain kinase, right, phosphorylates the myosin head, right, and then step 6 is myosin binds to actin, forming that cross bridge, right? And then we can just say cross bridge cycling, because the cross bridge cycling process is the same in smooth muscle as it is in skeletal muscle. Right, so what I want to do is I also want to put over here, right, just a small little arrow that says, we'll call this step like 4B, right, calmodulin regulates caldesmin and calponin. Okay? And we'll sort of leave it at that, because what this actually does and how this interacts with the myosin and actin filaments, right, can be a little bit difficult, a little bit not well understood, so we'll sort of leave that for the graduate level, right? But as long as we understand that there is some sort of interaction that happens there, that's okay. All right, so that is what we get to the state of contractedness, right? So now if we want to relax this smooth muscle, right, we have to do a couple things. So I want to put contraction over here so that we know these are the steps of contraction, right? We'll put relaxation over here so that we know these are our steps of relaxation, right? So the first thing we have to do is get rid of the signal, right? Get rid of signal. So we have to do that. We have to get rid of that signal first, right? Then we have to remove calcium from the sarcoplasm. So where are we going to put that calcium, right? We have to put it back into the sarcoplasmic reticulum and the ECF, right? So we have two places where we have to pump it, right? This is going to use ATP, right? So we have to use a calcium ATPase to do so. So it's going to be energetically expensive, okay? Now the next thing that we have to do is we have to deal with the fact that this myosin head is attached to the actin filament. So we have to detach that myosin head, right? But in order to do so, we have to dephosphorylate that myosin head. So what we're going to use is we're going to use something called myosin phosphatase. Myosin phosphatase, right? So myosin phosphatase will come in and it removes that ADP, an inorganic phosphate, dephosphorylates that myosin head. So we'll say myosin phosphatase dephosphorylates myosin. Okay, so we remove that, right? And so once that is dephosphorylated, the myosin detaches from actin, and our smooth muscles relax, right? So this enzyme here, this myosin phosphatase, not an efficient enzyme. It's actually pretty bad at its job. And because of this, when smooth muscle contracts, when it goes through these steps of contraction, right, this process right here, the action of that myosin phosphatase, not efficient, slow. So myosin is slow to relax. So I'm just going to put that up here so that we know, right? Slow to relax. Smooth muscle. Okay, and it's because of that myosin phosphatase and its inefficiency. Okay, so those are the steps of smooth muscle contraction and relaxation.